Welcome, everyone. Thank you for joining us for today's track on cell biology of diseases. It's my pleasure to introduce to you Dr. Marie-Christine Birling, Head Associate for the Genetic Engineering and Model Validation Department in the Institut Clinique de la Cerise. Since 2013, she has implemented the CRISPR-Cas9 technology, and now most of the models are generated through this approach. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Birling's keynote presentation is titled CRISPR-Cas9 Genome Editing in Mouse and Rat, the PHENOMIN-ICS Experience. My name is Antonina Pavlenko, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Marie-Christine Birling. I will now turn the presentation over to her.
Hi everybody, I'm happy to present you today the PHENOMIN-ICS experience. Just to tell where we are, because I think we work with CRISPR since a while, but we are the French mouse clinic here, and I'm head of the genetic engineering and genome editing service, where we do all this work about generating new mouse model. We are part of PHENOMIN, which is a three centers are related in PHENOMIN. That's the left panel, and we are also part of Cellmedia. And that's on the back, you can see the center and the ICS mouse clinic in Strasbourg, near Strasbourg in France, is on the left panel on the bottom. Okay, let's go now to CRISPR. To CRISPR, what we can do with CRISPR, I'm just that. With CRISPR, what we can do, CRISPR simply what it does is generating a double strand break. Then when we introduce CRISPR in cell, all CRISPR is doing is generating a double strand break. And of course, afterwards, what can occur if after the double strand break, the cell doesn't like to have DNA that is cut, and then it will kind of reglue this DNA, and that's called non-homologous end joining when there are some mistakes. Then they can be indels or small insertion in this case. But what we are really interested in is definitely something that is a specific correction, and that's done when we inject or electroporate or introduce Cas9 with CRISPR with the donor DNA. This donor DNA can introduce the mutation we want. And it's possible to use a single-stranded donor DNA or a double-stranded donor DNA. And this event is definitely the event that we would like to emphasize here. And we are not so interested in the generation of NHJ, but in generating precise event. Okay, then at PHENOMIN-ICS, we are using CRISPR-Cas9 not only to generate mouse lines, but also to generate rat lines, because CRISPR works on everything. Of course, in mouse, we have also ES cells, but here in the first instance, I will show you what we do with mouse and rat embryos, because we introduce CRISPR-Cas9 with or without a donor DNA in the embryos, in the one-cell embryos, or by microinjection or by electroporation. In the mouse, we have another approach, which is ES cells, but in the rat, it's the only way to obtain genetically engineered rat. I have to say that we are also part at PHENOMIN-ICS, like many centers in the world. You can see that on the top right panel. We are part of the IMPC. And the IMPC, the aim of the IMPC is to generate knockout of all the protein coding gene in order to do a high throughput phenotyping pipeline on all these lines that are generated. As part of this consortium, we have generated quite a few lines, knockout lines, directly using CRISPR-Cas9. And the approach that we take here is deleting a critical exon. This exon, when it's not there anymore, will generate, the deletion of this exon will generate a frame shift and lead to a premature stop codon, and so lead to a knockout. Of course, we also do these knockout models for collaborators. And I just want to emphasize here that it's really, really very efficient, because even by microinjection, you can see here that on 7 pups born after microinjection of CRISPR-Cas9 and 2 pairs of guide, we have more than half of the animals usually that have the deletion and that are potentially interested that can be bred to generate the knockout. Then it is really efficient and really cheap. And then the time needed to generate this line can be decreased to 5 to 6 months instead of going to more than a year going through ES cells. Then that's really interesting. And that's the pipeline that all the MPC is taking at the moment. It's fast, cheap, and easy. And the only drawback is, of course, that we only have a knockout allele and no reporter and no conditional allele. And then, as I said just before, that generates a straight knockout when you use CRISPR. But here I just want to emphasize that we, as part of the MPC also, we are also generating lines through ES cells, because there was a whole pipeline of people that were generating ES cells with a much more complex allele. And this complex allele is a knockout first, because it introduces a LAGZ before a critical exon, which stops the expression of the gene. And once you obtain the line, you can breed this line with a flip deleter line in order to obtain a conditional knockout line or a knockout line by removing the critical exon. Then this allele is still really highly valuable, and we continue to produce this mouse line, because as a standard, as a normal scientist, when they are really interested in a gene, they don't only want to have a knockout allele, but a more valuable allele that can be a knock-in or a conditional knockout, and then they can breed their line with their preferred CRI. Okay, and just to say here, I just add that most of the conditional alleles that are available at the moment are coming from the IMPC, because we have generated in this consortium more lines, more conditional alleles than in the whole world beside this consortium. And it's the same for the reporter allele. Then the work for this from this consortium is quite impressive, and it's really valuable. And you can go and see on the website that is stated at the top of the slide. There are many interesting data on this website. Go and see there. But no, what I want to emphasize here also is that we can also generate knockout in rats, and that works really well as in mouse, definitively, by injecting just 2 pairs of guide RNA with Cas9 into an egg and targeting a critical exon. This critical exon will generate a frame shift. We can generate knockouts. And these slides are really nice. I really like to show them, because you can see here, it's our collaborators that give them this slide. You can see the dystrophin that is really nicely expressed in wild type, whereas in founders, you can see that the animal that are obtained in the first instance are mosaics, and only some cells are expressing dystrophin. Then it's a really nice way to see it. Here, again, it's interesting because often the examples that are shown about the efficiency of CRISPR are used on this model of myopathy, because this gene, dystrophin, is located on the X chromosome. This means that when you work with male, it's much more easy than when working with females, where you have 2 alleles to target both chromosomes. In males, as it's on the X chromosome, you have only 1 allele to target. And here is the genotyping that we perform. F4, under the F4, it's the males that we genotyped first. Then you could see only one allele. Then it looked almost homozygous. And the animal named numbered 1 to 14 are the F1. And you can see that all the females had the deletion, which shows that it's really efficient. And this line is still not published, but it's definitely the first Duchenne muscular dystrophy model in rat, and will be certainly really valuable to study the model more in depth. Okay. Now, about conditional knockouts and CRISPRs. Then in my facility, I have often people that come and ask me, can you do conditional knockout really fast and cheap? And that's, of course, what everybody would like to have, because conditional knockouts are really valuable model to understand the role of the genes in certain, when bred with some pre-deleter lines. Then is it easy or not easy? That's the question. Then to remind you, I don't know if you followed all the literature, but in 2013, at the really beginning of CRISPR, there was this paper coming out saying, from Rudolf Jenisch lab, saying that everything will be simple. They showed that everything is efficient. It's possible to target 3 genes together, to get easily conditionals. But all what they did here, they were analyzing font loss. And what happened afterwards, it's that a lot of people tried to reproduce what was on the paper. Some things were reproducible and some other less. And that was the case for conditional. And in 2019, this year, a paper came out from Biography and a lot of different teams that says that it's not so simple to obtain conditional knockouts, simply because if you use 2 SSODN with both LOXP side on both sides and a guide RNA on both sides, you will get only very few successful projects. And the success rate was less than 1%. That's really bad. And why is it so bad? And that's certainly the reason why I didn't want to begin to try to get conditional knockouts using this method here with 2 single-stranded oligonucleotides. And that's again the case. The desired outcome is less than 1%, as I said. But what you will get? You will get a LOXP on one side, a LOXP on the other side, indels on both sides, deletion, and all these things can occur because you generate a double-strand break and you want to have some specific recombinations that occurs. And when you generate 2 double-strand breaks at the size of both LOXP sites you would like to introduce, you generate, in most of the cases, a deletion, because it's 2 different breaks, and that generates a deletion. Then it is not so easy to obtain conditional knockout. And of course, no, there was this paper in 2015 and 17 saying that the solution would be single-stranded DNA rather than double-stranded DNA. And there was this paper there from Murphy again. And they said that in this case, it will work. And here I want to show you one project that worked pretty well in our hand, saying it's not really obvious because we observe a little bit the same kind of things that was observed by all the people that try to do conditional knockout, but with a single-stranded donor DNA instead of 2 single-stranded oligo to insert the LOXP site, it will work better. And that's what we obtain. Then first, on the top, the drawing of the scheme of the experiment you wanted, we used 2 guides that are called GR67 and GR73. And they were microinjected with single-stranded DNA and Cas9. And that's the numbers that we obtained. We obtained 34 pups, and we got 1 pup that had both LOXP side. And that was done by performing a PCR from inside to outside of the construct. Then we were sure it was at the locus. We also obtained 1 founder, 1 animal that had a nice deletion. But we also observed, and that was already described, and that was described in between, we obtained 4 animals that had only the 5' LOXP side and 3 animals that had only the 3' LOXP side. But it's possible to obtain what we want because we bred heterozygous pups that had both LOXP on the same allele. We showed that indeed they were on the same allele, which is important, not on opposite allele, on different chromosomes. And when we bred them, you can here see really nicely genotyping. You can see that we obtain homozygous pups with a green arrow, and that indeed that's true, because even when we perform the PCR from outside to outside, you can really see nicely the shift. And we validated that everything is fine, and that is really important. We validate that by 1,000 rods, seeing that you have only one insertion, and that's with an internal probe, one insertion of our single-stranded DNA. And we also use droplet digital PCR. We put a probe as an internal probe, but a smaller probe by droplet digital PCR, and we have only 2 copies of the wild type allele. That was great. And on the homozygous, we were able also to fully sequence all what we introduced. And what can be dangerous with single-stranded DNA, I still believe it is the case on any project, is that you can introduce mutations that you don't master. And that's why perhaps double-stranded DNA is a better solution, because there will be less mutation there. Now, next point I want to emphasize here, point mutations, that is pretty simple and works really very well by microinjection or electroporation. We had plenty of successful projects. The only limitation is a dominant lethal allele, and in this case it's annoying because the founder will die, that we obtained after microinjection or electroporation of the Cas9, the guide, and the donor DNA. That can be a pain. And here I want to show you one example, because it doesn't show well. One example of a model that we try to reproduce. It's a model for Alzheimer's disease. It was a humanization of the rat APP. It was on rat, and we wanted to humanize the 3 amino acids that are in green here, and to introduce this mutation. We choose a guide. At this time, it was one of our first projects that is called 78. And the approach is on the right panel. You can see the donor DNA had the 3 mutation, and we introduced an HPA2 restriction site. Okay, and we mutated the PAM in order to make sure that when we have the correct mutation, the Cas9 won't be able to recut. Then we need to mutate the other PAM or the recognition sequence of the guide. And that's the genotyping we obtained. Here, you can see not all of the founder were positive, but when we digested the PCR product F1, F3, we could see that some founder had the correct restriction site expected. And here, I just want to show you the example of the founder 953. Here, if you focus on this one, it looks homozygous. When we looked at the chromatogram, because CRISPR is about looking very closely at the chromatogram when you analyze founders and even later. We spend a lot of time looking at chromatograms. And here you can see that even if the first mutation with the HPA2 side is really nicely looks homozygous on this founder, you can see that afterwards you can see little small double peaks. And that was intriguing. And even here, on the latest, you can see there is a real double peak. Okay? Then we bred this founder. We had the 3 mutation, we bred this founder, and that's what we obtained. It looked like all of the F1 pups had the mutation because the restriction site was there. But when we looked at the chromatogram again, we could see that only 3 of these pups had the 3 mutations. And the other animals had only one of the mutations that was on the HPA2 restriction site. That's why it is really very important to check really the chromatogram and check that everything is correct. And in this case, it was really important. Then our general observation is that the closest the distance between the point mutation we want to integrate and the selected guide RNA cutting site is, the better. We need to make sure that the Cas9 doesn't recut after HDR. Then in the donor DNA, you need to mutate all the PAM or the sgRNA recognition site in the donor fragment. If not, it will recut and recut, and you will never obtain the mutation you want. It is nice when you want to ease the genotyping to introduce diagnostic restriction site. And of course, this one needs to generate a silent mutation, because of course you don't want to introduce mutation in the coding sequence. There is one slight risk, is that introducing silent mutation could generate some strange splicing, and that needs to be assessed. Of course, when you do point mutation, the other outcomes are knockouts, because you might get also NHG, that's a small indels allele, and if you have a deletion of something that is not a multiple of 3, you will get a knockout, because it will generate a premature stop codon, and you can also get the knockout. And finally, and that's real for every founder that we obtain, the biopsy that we take from this founder, it can be ear, tail, or finger, does not completely reveal the genetic makeup of a founder. We really need to breed this animal in order to make sure that you will indeed get what we want. And by breeding these animals, we will really set up a line and be sure of the line we obtain. Now, I told you it's pretty simple to obtain point mutations. That's true. But here, I would like to show you a difficult case. That's a case, one case that we had, and I think it's nice to share it, because sometimes CRISPR is not, it's not so easy, because you need to have a guide that cuts specifically. And that was the case for a point mutation we wanted to introduce in this gene here, SCN10A. And you can see here, it's surrounded by red. This gene is here. But besides this gene, there are some other genes of the same family, SCN5A and SCN11A. And when you look closer at that, you can see that when you align both sequences, you can clearly see that the sequences for SCN5A and SCN10A are pretty identical. Then at the size of the mutation that is surrounded on the top, on the low panel, you can see here that you cannot choose a guide that is specific, because it will also cut in SCN5A. Then it's pretty complicated in this case to have something that is really specific. We tried this way. Then instead of introducing at the red side here, the mutation, because you couldn't get a specific guide, we tried with 2 guides, and we tried to introduce the mutation here with guides that were more specific from the alleles, and unfortunately we were not successful. And this is, no, I want to tell you that that was not very interesting. We couldn't get the line. That's why we decided to go through ES cells. And in the last part of my talk, I will re-speak about ES cells, because we still have a very good ES cell facility and good ES cells. But ES cells can still be really helpful. And in this case, it was because we were able, in this case, to find a guide that was completely specific, because it was in the intron. And this guide was put at the place of the insertion of the neo selection for this point mutation. And we were really easily able to obtain many positive clones and to validate this positive clone and to obtain germline transmission. Then we spend a little bit of time with CRISPR, but sometimes ES cells are still the correct way, because you can take a different approach with other constructs than just a single-stranded oligonucleotides, because sometimes it's not possible and you have no guides that are specific enough. In this case, we are completely sure that everything is fine and that we are at the correct allele and that you haven't targeted the SCN5A allele. And that, I think, is really a nice tool to still have, is ES cells with CRISPR. Then small knock-ins, just a few slides to tell you that that's also pretty good. That's one example. In this case, we had to introduce 3 times HA tag at the ATG of one gene. And you can see here the genotyping. We obtained many founders. I will go pretty quickly through these slides. Even some of these founders looked almost homozygous. That's founder 21, 22. And that was really nice. And we very easily obtained germline transmission from this line. That is really efficient. Then for small tags, that works pretty well. Then no problem with that. And now one thing that is a little bit of specificity, or something that we really developed at PHENOMIN-ICS, is a structural variant, because we know a lot of things that we can do with CRISPR-Cas9, and I think that's, even if it's published, it's not so well known, and you need to know, and you can play and generate much more fancy alleles with guide RNAs and Cas9. And that's why do we want to generate structural variants? That is because in the human, there are many diseases that are due to chromosomal aberration. There are some variable trisomies that concern mainly only, I would say, because if no, it's lethal, human small chromosomes, and mainly trisomy 21. That's the main subject of the director of the ICS, and that's why we are really interested in this model. But there are also some humans, some babies that are born with trisomy or chromosome 18 or 13. That happens. And of course, there are also many human diseases that are due to microdeletions or microduplications. And I cite them all here. I won't spend the time to describe them all, but anyway, they exist and they are there. Okay, then we need animal models to study these syndromes. And that's what we did. We generated new rat Down syndromes models. I won't go through the way most models were generated before CRISPR, but you need to know that they were using loxP-side. It was taking 3 to 5 years to obtain a line. And I will show you here that when we decided to go for the rat model of trisomy, of human trisomy, that's how we proceeded. You must know at the beginning that the human chromosome 21 is a small chromosome, which on the left panel here, has 2 main synthetic domains, and domains that contain the same gene. And these are on rat chromosome 11 and on rat chromosome 20. Okay, then if we duplicate the domains that are on rat chromosome 11, we will have a model that is close to trisomy of chromosome human chromosome 21 for this region, and the same for rat chromosome 20. Okay, then that's how we proceeded, and that's an example of this region, the first region here that is on human chromosome 11. And here you can see this region is already quite big. It is 3.6 megabase. And this is the approach that we undertook. We undertook this approach, again, like doing knockouts. We used pairs of guide RNA simply because we thought if one guide doesn't cut well, the other one will cut. And as you want to delete something that is large, it would work. And we designed primer all over the regions to be able to check if we see inversion, duplications, or deletions. Okay. And in the case of this region here, that is a region that is synthetic to human chromosome 21, that's what we did. We obtained animals. We couldn't see anything by junction sequencing. That's why we we ordered probes for droplet digital PCR, and that's on CBS. That's in blue. You can see it in blue here. And we performed droplet digital PCR on some of these founders. And you can see here, on the left panel here, you can see the genotyping, the droplet digital PCR results of the first founder we obtained. And you can see here that we have one founder in which we could see the deletion, at least of CBS, but there was most probably the whole region was deleted because CBS is in the middle of this region. And as we couldn't see duplications, because that's the model we are looking for, we bred the animals that were that are called here 801, and 802. Sorry about that. And we bred them. And we were really very pleased when we see that breeding this founder 802, we could see that in the first litter, we could see animals that had the deletion and animals that had the duplication. That means that this animal was mosaic. It had one allele with a deletion and one allele with a duplication. And then when bred, we could obtain all the duplication or the deletion, because after meiosis, you can only get one of the allele. And that was really nice. We could obtain that. And when we checked here with different probes, the different colors are also, you can see the gene on the top. When we checked with different droplet digital probe, we could indeed see that the whole region was or deleted or duplicated according to the F1 animal we could see. And thanks to that, we could look for the junction, because you can see that sometimes we observe that sometimes the deletion is a little bit larger than what we expect, and then we need to search for the junction and take oligonucleotides that are a little further than the real expected size. Then that's what was really useful, and this means we were able to obtain the duplication and the deletion of this line very fast, in a very fast way, and it was also quite cheap to obtain that. Then what did we learn from that? That it is possible to generate monosomy and trisomy and more models in a diploid context, and we have generated up to a duplication of up to 24 megabase, and that's huge, but it really works really well. And the only problem, the only limit to this approach is the allele lethality, or the ability to breed of the founders, because sometimes they are sterile, because of course we delete a lot of genes in some cases. Then what we also learn is that sometimes when we use primers on both sides of the deletion, sometimes we don't immediately see what we want, what I showed you before, because sometimes the deletion is much larger, and that's why droplet digital PCR really helps us to find the correct animals, and after we can find the junctions. And I think both approach, junction PCR and sequencing, and droplet digital PCR, are really great to help us to understand what we get. And it is really very fast, because we can get a line in 6 months, at least duplication or deletion, whereas before it was taking quite a very long time. That was published, and it is really helpful. You need to know that. Then how far can we get without ES cells? Okay, then just to recapitulate here, what we can get, it is very easy to obtain knockout, indels or deletions and duplication, even if duplications are less frequent. It is easy to obtain point mutation and small DAG. Conditional knockouts are still difficult to obtain, but it is possible. And no large knock-ins are still a problem, I have to say, with CRISPR, because it is not so simple to obtain them. And we don't really know why it's more complicated to obtain a large knock-in instead of a small knock-ins, but the mechanisms are not exactly the same. That's what we observed in ES cells, definitely not. Then EasyCRISPR can help. EasyCRISPR is using a single-stranded DNA, that's what I showed you before, but it's error-prone, and you can have mistakes in the donor DNA, and that can take a lot of time to fully validate a line, because you need to resequence everything. And here I want to say, when you do a large knock-ins with CRISPR, you need really to check the whole picture. That's why I put this picture of the parable of the blind man and the elephant. Don't just look at one thing. You need to get a whole picture. And I think that you need to add droplet digital PCR, or even better, southern blot that will give you the whole picture of your allele, because if you just do small PCR, you might miss some event. And I really believe that in the world, there are many lines that people believe they are fine because they did only small PCR. And if you would look much closer, you would see that everything is not completely fine. Then really be careful and look at the whole elephant, at the whole picture, instead of just at one little test that is sometimes not enough. And now, to finish this little part here, I want to say that there was this paper coming out from Janet Rossan, 2C HR CRISPR, which seems to be really interesting, and this experiment seems to work already in some labs. We are testing that at the moment at the ICS, and this method was first published in bioRxiv and in 2018 in Nature Biotech, and it takes the advantage of the long G2 phase and open chromatin structure in 2-cell stage embryo. Okay, and instead, when you inject in the 1-cell stage, you injected the S stage. With the 2C, you inject at the G2 stage. Then this technique is supposed to work, and works in some labs with double-stranded circular DNA, in which you can completely sequence and be sure of the sequence. And I think that will help to generate efficiently large knock-ins. And at the moment, we are testing that, but we have still no data at the moment. but I think it's a good future, perhaps certainly better than single-stranded DNA. Now, I just want to show you the benefits of electroporation that we developed, like a lot of teams, of course, but I think it's nice to share also in this webinar, is to tell you that electroporation is really nice. And our data, first, show you microinjection, needs some really skilled people because you need to go into the pronuclei. Some people like to inject into the cytoplasm, but in our facilities, they don't like to inject in the cytoplasm. You need to have very skilled people, whereas electroporation is much simpler. You put your eggs, your fertilized egg, in between two electrodes, you pulse with this square pulse. We use an EPA21, but all the electroporator, will work. And just to say, first we checked by using mRNA for GFP, and you can see here that it works really well. You have nice 2-cell embryos that are very nicely GFP. And that's what we did. We tested a few projects here, 5 projects, and that's the data we obtained. They are the same project with microinjection left panel or electroporation right panel. And you can really see here that even if you obtain approximately the same number of animals, you can really see that the number of positives that are the orange column are much, much higher when we use electroporation, because you have not anymore the bias of the microinjection. And that is really great because you increase the viability of the embryos, and then you need to inject less embryos into foster mother, and it is really nice. Then it's really interesting for the 3R because it really reduces and minimizes the number of animals used per experiments. And what is also really interesting is that it really reduces the cost because we need to use less animal, less donor female, less foster mother. Then electroporation is really something that we are using now for all of our projects. Then we have done not so many, but they all worked really nicely. On average, I think we don't need to reimplant more than 3 foster molar. That's about 60 eggs, 60 electroporated eggs to get 2 liters. And in these 2 liters, we have more than enough founders to breed afterwards. What is really also very nice with electroporation is that we can just buy 4 or fertilize oocytes from Janvier, for instance, in France. We can get them and we sow them, and then we can just electroporate these oocytes, this fertilized oocyte, without having to breed animals and so on. That's really, really convenient. Also, if you don't have too many projects, you can just throw straws and work on this one. And of course, it opens the field. It should work in any genetic background. It works also really well in B6J genetic background. All of the projects I show you in mouse are done in B6N genetic background. Sometimes we need to do that in J. But of course, it also works in BALB/C and so on. And that's really nice. We also know, as we are really confident in our electroporation, what we do know when we don't obtain positive, we will just do an electroporation of approximately 40 embryo, go them through blastocyte, and check if we have what we want. And if we get what we want, we can say that afterwards, after reimplantation, that means that the embryos are dying because the modification we do is lethal. And that's really nice because it validates the approach, and that's really nice. And that's how we do it. And I have to say, approximately the same method works also with rat electroporation. We plan to write a publication saying you don't need to develop a completely different approach. It really works well for rat oocytes too. Now, another part I wanted to emphasize here is RNA-guided base editing. Then that's another CRISPR toolbox. And it was quite interesting, and some people asked us if we don't do that. And here I want to tell you, I want to share what I think and what we obtained doing some tests with this RNA-guided base editing. Then what is RNA-guided base editing? Simply, we know that Cas9 is a wonderful protein because you can fuse it with any protein you want. And in this case, you can fuse this with a deaminase, and it still does its function when it is with a guide RNA, it will generate this complex, and the complex will recognize the sequence that you want to target specifically in any cell type. That's the way how it works. When we use RNA-guided base editing, usually the Cas9 is or dead Cas9 or nickase Cas9. Okay, then that means it is not supposed to generate double-strand break or to generate break. It's just supposed to sit there and to do what I will show you, deamination. And if you use cytidine deaminase, it will change a C to a T. Okay, and that's what it will do. You can, and sorry, that's what it will do. And afterwards, how it does work, cytidine deaminase is great because it's linked to this deaminase, it's called ApoBec1, and it has a specific editing window. As it doesn't cut, ApoBec will just modify the cytosine that are located, in theory, in this editing window. That is not really precise, but that is presented as being an editing window that is located 12 to 16 base pair from the PAM sequence. Okay? If you use cytidine deaminase, the principle is exactly the same when you use adenine deaminase, but it's a little bit more complicated because they had to fuse two domains, but still that's the way it works. Then, how they present that in the papers, they present that there is a real advantage. There is no need to use a donor DNA. There is no double-strand break, so no deletion. That's what I'm asking. That could be really especially interesting for gene therapy or cell modification for therapy, because of course, when you use CRISPR-Cas, everything is afraid about off-targets and so on. And the cons is that it is not precise enough. The real problem is that in this editing window, if you use cytidine deaminase, if you have more than one C, it can potentially modify all the C. And of course, you are limited in the mutation you want to introduce, C to T, G to A, according to the strand on which you are, or for the cytidine deaminase, or A to G or T to C, according to the strand for the adenine deaminase. And also, of course, if you have off-targets and if the guide is recognizing another region and you have a C for the cytidine deaminase, it could potentially also modify something else. And that's an example that was published, and it's a Nature Biotech paper here. You have the reference here. And in this case, they use the adenine deaminase. You can see here that the A can be transformed in a T. That's with on this model of dystrophy, okay, again. And in this case, they use AAV, introduced adenine deaminase, and with a guide RNA that is specific, that is underlined here, that you can see here. And they said they obtained on male founder, of course. They were really happy to obtain on this model that was a mutation that we wanted to correct the mutation. Okay, they obtained, whereas the animal at the beginning was a knockout, they could observe with the base editor that some cells, muscular cells, are again expressing the dystrophin. That looks great, but of course, if the correction is not complete, you can also generate a deletion, and it can still go back into the frame and generate a dystrophin that is almost normal. Anyway, they say that works really well. Again, it's on a model that is on the X chromosome because it's simpler and because you need to target only one allele in the male. And that's why this kind of model is often presented, because it's the simplest model possible. So we decided just to go and tested base editor, cytidine base editor, and on tyrosinase, and I will show you one, and two versions of this base editor 3 and base editor 4. And that's what we did. Here, we use this paper here. You have the reference, it's a Nature Biotechnology paper, and you could see that that's the approach that they did. For tyrosinase, what they wanted is to generate a stop codon. Tyrosinase, if you mutate tyrosinase, you should have animals that are not black anymore if you work on black 6 animals, but they go white if you target both alleles. Okay? Then base editor, it goes from a C to a T by generating a stop codon. Again, I remind you what is published, the editing window. And then we took the same guide RNA generated DNA. First I show you what they showed, and they showed that indeed they obtained the T. Some look correct, but you can already see here that some mutations are not the ones that are expected, okay? And that there is not a C, but it's transformed to a G in the paper. And sometimes you can see also in the T112 that you can have indels, which is not great if you think you would use that for therapy, I think. And they say, okay, in embryo, they see that animals have white eyes, then they should be okay. That's fine. And what is even more surprising is the conclusions saying that you can indeed, this approach would be useful to introduce single amino acid substitution and correct genetic defect in human embryos in the future, which is surprising because when you see what you observe at the top, I think you see something that is not very specific and that didn't look so much more specific than normal CRISPR-Cas9. So what we did, we ordered from Aging the plasmid, coding from BA3, citidine deaminase version 3, and citidine deaminase version 4 that were published, one in 2017 in Nature Biotechnology, the other in 2018, not in 2018, they didn't test that in vitro, they just tested that on cells, but it was published and they say this base editor 4 is better than the base editor 3, the third version. And that's what we did. Then we didn't go until generating this animal for 3R region, but we generated the guide RNA, the RNA for base editor 3. We obtained first animals. And after what we obtained when we analyzed blastocytes, no, that's just on animals. And when we analyze these animals, you could see clearly on these animals that it's really surprising because I put the correct modification, expected modification in green, and all the other modifications are in red. And that is definitely not very specific. Then it's a little frightening to see that. And I think I wouldn't trust that to do genetic therapy. But that's the base editor 3 version. And now we can go and see what we obtain with the base editor 4 versions, that's genotyping blastocytes only, not generating animals. And you can see here that it looks a little bit more specific, but still, even if the Cas9 was nickase, we can still see deletion than in the first paper that was published in Nature Biotech. And we can still see that the editing window is much larger than what is supposed to be. Then I think for therapy, we are still not there. But even more than the conclusion for this experiment is that definitely in mouse, even if it works, it is not great. I mean, when we sequence, we could see the conversion, the expected conversion, C to T, but we could observe also C to A and C to G conversion in most of the mutant blastocytes. We could observe deletion, insertions also in some blastocytes. Then that's no good. The editing window is much, much larger than what is presented first, even if it said it's not precise. Here it's 10 base pair. Then to generate a specific mutation, it will be very difficult. And what is more annoying is that, again, the animals are really very mosaic. We see many alleles, 2 to 3, which is not better than what we obtain with a single donor DNA, in which we can really only introduce the mutation we want and select for it. Then what I address here is that perhaps we could have mutated the Cas9 to dead Cas9, and perhaps we wouldn't see indels, but that we won't test again because honestly, I'm not convinced that base editing is the future of CRISPR-Cas9. For the moment, it still needs to be more precise and be ameliorated at the moment. Okay, that's all for base editing. And now, more or less to finish, I want to show you, I showed you already one example about CRISPR-Cas9 in mouse ES cells, because we use CRISPR-Cas9 also a lot in mouse ES cells, and that's to generate very complicated complex allele, because these complex allele, I told you, are very difficult to obtain by microinjection reconstitution into eggs. That's why we still use mouse ES cells. And that's our data. That's just to show you all the projects numbered here, 1 to 23. And here you can see that the number of clones that we analyzed first, and some of these projects are really old, and that's why when CRISPR came out, I thought, okay, we didn't manage to obtain one clone. For example, the project 3, we analyzed more than 1,500 clones without any positive clone. And then I just decided to use the same targeting construct, putting one guide, generating a double-strand break at the side of insertion of the neo, and you can see here the number of positive clones that you obtain. It's really a lot, and it's quite amazing how a simple guide RNA with Cas9 can change the number of positive clones we obtain. And that's definitely really important. It was efficient really with linearized construct, with long and short arm for the same construct. It was very, very efficient. Then that also means with CRISPR, we don't need, when we target ES cells, to clone big, large homology arm, but we can use short homology arm. 500 base pair is more than sufficient. That's really nice. What we observe, and that's what we always still do, thousand blot to really check, we observed concatenars in a lot of these clones. Even if we manage to get easily what we want, you have concatenars, and that's something I'm trying to explain here, is even if you have homologous recombinations that occurs on both sides, we have more than one copy that is inserted at the locus. Even if the 5' long-range PCR and the 3' long-range PCR is fine, in the middle, it's not correct. This means that if you use an internal probe, you will see more than one band. And that's, again, what I want to say. I, again, show my little elephant with the blind man. You really need to check what you obtain when you use CRISPR. Be aware that you can observe sometimes some strange things, and check that everything is fine. And that's an example because, of course, complicated models are feasible easily with CRISPR, but here it's a model that we have generated, and that's even larger. Then it's possible to do humanization pretty easily using ES cells simply. And that's a model we have generated. In fact, 3 models with the 3 mutations that you can see in green, blue, and red, ApoE3, ApoE4, and ApoE3, 2, and 4, okay, with the different mutations here. And how we proceeded for that, we worked with Gene Bridget for the construct. They inserted everything that is on the square here from a human back. They cloned homology arms around. I show you the construct here. I don't know if you can see it well. Everything that is in blue on the construct, on the circular construct here, is human, and you can see in green the homology arms, and they are here. And we cloned at ICS plasmid with the 2 guide RNAs that were located at the size of the insertion we wanted to do. And when we performed the electroporation, that's what we obtained. We really obtained a lot of positive clones doing long-range PCR both on 5' or 3'. This means that homologous recombination worked well. And that actually also worked well if we used only a selection pressure, because in the targeting construct, we had 2 selection markers, one hydro selection marker that was for the 5' insertion, and one neo selection marker for the 3' recombination. And using a selection neo-hydro or neo-hydro, we obtain positive clones. And sometimes, of course, we also obtain these famous concatenars I was talking about just before. And just to say, that's how we validated our lines afterwards. We used a neo-probe and a hydro probe. You could see that in most of the cases, we had only one bond, which is nice. You can see that in thousands, that's why I want to show you here a plot, that with a little arrow, you can see the expected bond, but also what is sometimes disturbing when you work with CRISPR on ES cells and you do 1,000, you can also see that the wild-type allele can move, because you can delete completely the wild-type allele, and you can generate the deletion of the whole part on the other allele, even if the targeted allele is correct. But as when we see the correct targeted allele, we don't care about the wild type allele. And that's very important also when you analyze ES cell clone that are issued from CRISPR Cas9 electroporation. Then we confirm that the humanization was correct. Everything is there. We humanize ApoE, Tom40, and ApoC because it's what our collaborator wanted to have. And I have to say, with the three versions of ApoE, we obtained germline transmission without any problems. Then it was pretty easy. And even if we used ES cells in a year, we obtained the 3 lines. And it's not so easy to obtain that through CRISPR. Then we obtained the lines. And that's just a review. You can go and have a look at the review we wrote here. But just to say that it's supposed to work with bat plasmids because it was published in Nature Communication in 2016. They showed different ways to obtain humanization, but that worked only in rat. I don't know why, but I think a lot of people tried to obtain the same approach using back with 2 guides, and they call this 2 hits, 2 oligo, or 3 hits, 2 oligos. They tried this approach. You can go and see. I won't go into the detail of this experiment. It seemed to work in rat, perhaps because of the eggs. The rat eggs are more resistant, but when you go into the mouse, it doesn't seem to work so well. And then in C, homologous recombination in ES cells allows us to obtain the same kind of thing in more or less the same timing, perhaps a little longer, but at least we can validate everything very well in ES cells, whereas you cannot do that if you obtain directly animals, and you have to breed animals. Then, as I told you, that's just speaking about kind of humanization we can obtain. That can also address a question: what is the best humanization? It's really a question we can now address because we have CRISPR-Cas9. Is the introduction of a specific causal mutation a better model than the whole gene humanization? No, we can humanize the whole gene. Then what is the best model? What do we have to humanize also? Do we have to keep the human regulatory sequence or just replace from the ATG to the stop? That's also a question we can address. Is it really important to keep all the human intron, or is it better to use a cDNA for trustful expression of a gene? Because when you put human intron, will they behave the same than in rat? That's really a question you can ask yourself. And also, are humanized protein interacting as efficiently with their rodent counterpart? Because is it a good idea to humanize a whole gene and put it in a rat or a mouse? I mean, sometimes you can imagine that as they don't have their real interactors, and perhaps it won't work as well. But anyway, these are questions I like to ask to people, and it's a nice way to think about it. Okay, at least we can do everything. I think everything is now possible with CRISPR. It really has rendered everything quite easy to obtain. And how far can we go with CRISPR? Of course, there is still a lot of space for improvement. There are improved Cas9 variants. I have to say we haven't tried them so much because Cas9 works so well in embryos that it's not really worth it. And also, what I wanted to emphasize here, we are not caring so much when we work on animals about off-targets, because we go through meiosis, and meiosis is a wonderful reshuffle of all the genes, whereas if you work only on cell lines, you will never get rid of off-targets. That's why people working on animals are not caring so much, because when you generate a line, you breed your founders, and you will segregate your off-targets if it's far enough genetically. And that's really why we are not so much into studying the different variants, but there are alternative proteins, CPF1, CPF2, and that this one can be interesting. We would like to minimize mosaicism. That's still a problem, even with base editing. I showed you that very clearly. We would like to reduce off targets. Then perhaps we could decide to dose amount of Cas9. Sometimes when it's too efficient, perhaps to get less events, we'll get an embryo to give a born pups instead of putting too much Cas9 and generating just a dead embryo. And of course, there are people working on inhibitor of NHJ, modulator, and so on. There are many things and many teams that are working the way to improve CRISPR-Cas9. Okay, and now to almost finalize what I want to say, CRISPR-Cas9 is great. I showed you that. But I think you need to really think carefully, at least at our place, what you want to do. I put a scale, the time in months, and you need just to realize that the time to fully validate an allele when you are working with ES cells, it takes the time of the cell growing, and you can do plenty of PCR on your clone, check that everything is correct, and you will spend your time validating everything in ES cells and only inject one clone that is really perfect, and it's really great when you do big modification, large modification. Whereas when you use CRISPR, you can go fast, you will obtain very fast, very quickly a founder, even germline, but after, remember my little elephants, you still need to really validate that everything is perfectly fine. I think it's really important to keep in mind that even if CRISPR looks really simple, everybody wants it to be really simple. It is simple, it is great, wonderful, but you really need to validate carefully the lines that you are generating in order to make sure that you are not following something that is an artifact. And finally, just to thank all the people, that's my lab in which we do more or less all the CRISPR-Cas9 work with two people, Romain Lorenz, Laurent Scheffer, and Valérie Herbst. And the people that do droplet digital PCR, I want to thank Guillaume Pavlovic, Loïc Lindner mainly, and all the people that are involved, the ESL culture, because they do a great ESL work, Marie Wattenhofer and Benjamin Eisenman, and all the people from the microinjection, and all the responsible from PHENOMIN-ICS. And thank you very much. I will be happy to answer your question, and I hope I was clear enough. Thank you.
Thank you, Dr. Birling, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Dr. Birling will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.